Alrighty, here we go. I think this is part 4 of the final. Very lengthy. We're talking about leukocytes, white blood cells. They're involved in innate and adaptive immunity. The innate are the granulocytes and the macrophages. The granulocytes contain granules and include the neutrophils, basophils, and eosinophils. The innate granulocytes and macrophages are polymorphonuclear. The neutrophils are essential blood phagocytes. They're active engulfers and killers of bacteria. They kill with toxic agents located within their granules. They have a multi-lobed nucleus and they're the most numerous, those neutrophils are. Basophils function in inflammatory events with the release of histamine and allergies. And eosinophils, they're active in worm and fungal infections. They're also involved in inflammation and allergic response. Natural killer cells we'll get to later. Mast cells are specialized tissue cells similar to basophils that trigger local inflammatory reactions and are responsible for many allergic reactions. Phagocytic mononuclear leukocytes would be the monocytes and the macrophages. That would be the phagocytic mononuclear leukocytes. The monocytes, blood phagocytes that rapidly leave the circulation and mature into macrophages. They ingest dead cells. And macrophages, they're the largest phagocytes that ingest and kill foreign cells. So remember, the granulocytes are the neutrophils, the basophils, and the eosinophils. The neutrophils are the most numerous, multi-lobed nucleus. They kill with that the granules that release toxin. T helper, TH, or helper T cells, or CD4 plus cells. That would be the TH. They're also called helper T cells or CD4 plus cells. These are cells that are stimulated. They're stimulated by the antigen Ag plus MHC2. There are two cells of TH cells. TH1, they induce cell-mediated immunity, T cell, and TH2, which regulate antibody-mediated immunity or humoral immunity. Blah, blah, blah. B cell, remember the B cell that needed help. That was the TH2. The TH1 is cell-mediated immunity, and the TH2 is antibody-mediated immunity. Then you have your TC, which are the cytotoxic T cells, or the CD8 pluses. They are also stimulated by the antigen plus MHC1. Excuse me, I forget what MHC stands for right now, but I'll remember it here in a minute. What happens is a virus infects a cell. The viral proteins are broken down into peptides. The peptides bind to the MHC1, which is then displayed. The TC with receptor specific for that peptide binds with the infected cell. The TC secretes perforin, and the cell is lysed. Controlling microbial growth. You have microbiocidal versus microbiostatic. Biocidal is the killing of specific groups of microorganisms. Biostatic is inhibiting the growth of certain microorganisms. The microbial death rate, it's the decimal reduction time required to kill 90% of the cells. The heat treatments depend on the type of organism and the temperature, etc. Sterilization destroys all microbial life. Talked about this once already, so I'm not going to go through a lot of this again. Yep, that's okay. Physical controls. Cold does not kill the microorganism, it only slows their growth. Many die slowly. Filtration physically removes microorganisms. You have the mechanical method used to prepare liquids that can't withstand the heat, including serum and other blood products, vitamins, antibiotics, and hormones. And you have air filtration. means of removing airborne contaminants by a HEPA filter. Then you have drying. Vegetative cells exposed to normal room air gradually become dehydrated or desiccated. Lyophilization, I think that's spelled wrong. Lyophilization, combination of freezing and drying, and then osmotic strength. Salt and sugars are used to damage cells by plasmolysis, used in preserving foods. Chemical controls. You have antimicrobial agents. They have chemotherapeutic agents used to treat diseases, germicides and germostats, which are disinfectants and antiseptics. The selection depends on whether it damages the tissue or the object that you're trying to get rid of the bacteria, if it controls the target organism, and the purpose of the treatment. Testing germicides. Germicide is placed on a paper disc. The disc is placed on agar seeded with the microorganism. After inoculation, observe the zone of inhibition. It will not go around the disc. It will appear clear. 4 types of hypersensitive reactions. The type 1 is an allergic reaction. It's immediate onset, like hay fever, asthma. That's called atopy. And then anaphylaxis is systemic and sometimes fatal. Immediate onset allergies involve contact with allergens, antigens that only affect certain people. Susceptibility is usually inherited. On first contact with allergen, the bee sting, for example, specific B cells react, that's the cellular response, with the allergen and form a special antibody called an IgE, which affixes by its Fc receptor to mast cells and basophils. That's going to be on the test too, I believe. The B cells, let's go through that again. Immediate onset allergies. On first contact with the allergen, a bee sting, for example, specific B cells react, that's the cellular response, with the allergen, and they form an antibody called an IgE. This IgE affixes by its Fc receptor to the mast cells and basophils. This exposure to the allergen is the sensitizing dose that primes the allergic response system. Upon subsequent exposure with the provocative dose, the same allergen binds to the IgE mast cell complex by its Fab portion. So, the first time the Fc receptor binds to the antibody, the IgE antibody, the second time you got the provocative dose, the IgE antibody that's already bound to the mast cell by that Fc receptor complex. Let me rephrase that. The IgE, the same allergen, binds to the IgE mast cell complex by its Fab portion. This causes degranulation, that's the release of the intracellular granulars, histamine, prostaglandins, and leukotrienes, with physiological effects such as vasodilation, bronchoconstriction, hives, nausea, vascular permeability, and smooth muscle contraction. Diagnosis of an allergy can be made by a histamine release test on basophils and eosinophils, serological assays for levels of IgE, blood tests to look for elevated levels of tryptase, enzyme released by mast cells, and skin testing, which injects allergens into the skin and mirrors the degree of reaction. Short-term treatment, drugs that block the release of histamines, leukotrienes, or other inflammatory mediators, epinephrine in an emergent situation. Long-term treatment is desensitization. Therapy involves the administration of purified allergens to prevent reactions between allergen, IgE, and mast cells. Inject an allergen, stimulate the formation of high levels of the allergen-specific IgG, which have a high affinity for the allergen instead of IgE. These IgG blocking antibodies remove the allergen from the system before it can bind to the IgE, thus preventing the degranulation of the mast cells, which results in prolonged immunization. The antigen is administered in increasing dosage. That was the type 1 allergic reactions, immediate onset. Type 2 hypersensitivities, reactions that lyse foreign cells, cytotoxic antibody mediated. Type 2 reactions involve the interaction of antibodies, foreign cells, and complement, leading to lysis of the foreign cells. These antibodies bind to the cell, either their own cell or other cell, trigger a normal response that alters the function of the cell, induce phagocytosis, activate the complement lysis and agglutination. In transfusion reactions, humans may become sensitized to special antigens on the surface of red blood cells of other humans. That would be type 2 hypersensitivities, reactions that lyse foreign cells. Examples are the autoimmune system, antibodies bind to their own cells. Graves' disease, the antibodies attack the thyroid cells. And then Goodpasture syndrome, antibodies bind proteins in the kidneys. You can also have in type 2 hypersensitivities transfusion reactions. The ABO blood groups are genetically controlled. The A blood has A antigens on our red blood cell. Type B has B antigens. Type AB has both. And type O has neither. People produce antibodies against A or B antigens if they lack these antigens. So type A produces B antibodies, type B produces A antibodies, etc., etc. And type O produces both A and B antibodies. Hemolytic disease of the newborn. The Rh factor is another red blood cell antigen that becomes a problem if an Rh mother is pregnant with an Rh positive, Rh negative mother is pregnant with an Rh positive baby and becomes sensitized. First pregnancy poses no problem for her baby. She then makes antibodies against the Rh factor, and if she becomes pregnant again with an Rh positive baby, these antibodies can cross the placenta and attack the fetus. Prevention involves therapy with Rh immunoglobulin, RhoGAM, injection, which removes Rh positive from mother's blood during the first pregnancy. This injection is given around 28 weeks and after delivery. This antiserum reacts with fetal red blood cells that have escaped into the maternal circulation, thereby preventing the sensitization of the mother's immune system to the Rh factor. The anti-Rh antibody must be given with each pregnancy that involves an Rh positive baby. Type 3 hypersensitivities, immune complex reactions. Exposure to a large quantity of soluble foreign antigens, circulating free-floating antigens, stimulate antibodies which then bind to these antibodies and produce small soluble Ag-Ab complexes. These immune complexes are removed by the macrophages or are trapped in various organs and tissues, which incites a damaging inflammatory response. In an immune complex disease, these complexes are so abundant that they deposit in basement membranes of epithelial tissues. In response, neutrophils release lysosomal granules that digest tissues. Complement is activated and results in inflammatory damage. Examples of type 3 immune complex diseases would be systemic lupus erythematosus and rheumatoid arthritis. And then finally, type 4 hypersensitivity, cell-mediated delayed reaction, a delayed response 12 to 48 hours after exposure to the antigen involving the activation of and damage by T cells. This is the only type of hypersensitivity that does not involve B cells. The type 4. First exposure, the Th1 cells become sensitized and proliferate and become TD cells. D is for delayed. Second exposure, the TD, which is delayed, is activated by the same antigen and releases lymphokines and inflammatory cytokines that recruit various inflammatory cells into that member. This would be contact dermatitis. The buildup of fluid in cells at the site gives rise to a red papule. An example would be contact dermatitis. Examples of type 4 cell-mediated reactions would be poison ivy, TB skin test, organ transplants. So Microorganisms. Most are pathogens. Prokaryotic. They have no nucleus or organelles. They're very simplistic, like bacteria. Eukaryotic has nucleus and organelles like mitochondria. It includes fungus and animals. And acellular is not even a complete cell. Most viruses and prions, like those that cause mad cow disease, are acellular. 6 main types of microorganisms: bacterium, which is prokaryotic; fungus, eukaryotic; algae, eukaryotic; virus is acellular; protozoan is eukaryotic; and a helminth is eukaryotic. So the only prokaryotic is the bacterium. Gram-positive cell walls are thick. They also contain teichoic acid and lipoteichoic acid. Looks like a thick slice of cheese on a cracker. The cell wall plus the cell membrane, it stains purple. Gram-negative cells are thinner and sandwiched between the outer membrane and the cell membrane. Looks like a Nutter Butter stained pink. So, gram-positive cell walls are thick. Gram-negative cell walls are thinner, and they're sandwiched between the outer membrane of the cell, the outer membrane and the cell membrane. And gram-positive cell walls are thick, also contains teichoic acid, the cell wall plus the cell membrane. Structure of a fungus. Morphology. 2 basic morphological types: yeast and hyphae. Yeast are round or oval shaped. They're single-celled. Grows swelling on its surface called buds, which then become a separate cell. Dimorphism means 2 shapes. Switch between growing as a yeast or a moldy yeast grows at body temperature. Mold grows at room temperature. So, example would be histoplasma capsulatum, causes histoplasmosis, and Candida albicans. Hyphae, they're individual filaments. Mycelium are connected hyphae, they're molds. Mycelium are molds. Lower fungi, if the walls are undivided. Higher fungi, if the mycelium appear to be divided by a septa. Can also have a fleshy body, a thallus. Fruiting structures, mushrooms. Structure of a segmented helminth. Tapeworms are made up of the scolex, which is the head of sorts with suckers on it for attachment. The proglottis, which is the body segments, and the germinal center, where new segments are made. Streptococcus pneumoniae, known as pneumococcal meningitis, affects weakened individuals. Pneumococcal pneumonia caused by Streptococcus pneumoniae. A peptidoglycan is an example of a polysaccharide. It's a combination of a protein component and a sugar. Bacteria. Prokaryotic true bacteria has a peptidoglycan cell wall. Provides shape, helps withstand turgor and pressure. Made up of chains of alternating polysaccharides. The NAG and the NAN. Cross-linked with peptides. Terminal electron acceptor drives the electron transport. The terminal electron acceptor divides the electron transport. If aerobic respiration, the final electron acceptor is oxygen. If anaerobic reception, the final electron acceptor can be sulfates, nitrate, or fumarate. Endospores, sporulation, occurs when conditions are unfavorable for growth, like extreme heat, cold, dehydration, presence of toxic chemicals, or radiation makes for long-term survival. Spores can survive for hundreds of years. The process, its unequal cell division begins. The cytoplasm divides into the vegetative mommy cell and the forespore, the baby spore. Each contain DNA. The thick protective wall forms around the baby spore, made up of peptidoglycan that is different than the vegetative cell. It is keratin-like as to its hardness. The spore contains all essential cell components. The vegetative cell lyses and releases an endospore. The endospore then germinates when conditions become more favorable. Histoplasmosis, also known as Ohio Valley disease. It's by the respiratory infections caused by fungus, histoplasmosis. Dimorphic means two shapes. Moldy yeast grows at body temperature. Mold grows at room temperature. Chlamydia life cycle. Transport into the cells of nutrients. From here on out, this info is based on looking at E. coli as our model. Outer membrane. Remember that E. coli is gram-negative, so it does not have an outer membrane. Nutrients pass through a poron, a small pore, down a concentration gradient from areas of high concentration to low. The cell wall is mesh-like and therefore nutrients diffuse through. The cell membrane can be hydrophobic. Molecules pass through. Hydrophilic molecules need transporters. Oxygen reduction reactions. Oxidation is loss of electrons. Reduction is gain of electrons. The end products of glycolysis: 2 pyruvates, a 3-carbon molecule with no phosphorus, 2 NADH, and net 2 ATP. Created 4, but it took 2 to make. So you get 2 pyruvates as the end product, you get 2 NADH, and 2 net ATP is the end product of glycolysis. The pentose phosphate pathway, alternate routes in glycolysis, production of necessary precursors, a ribose or an erythrose, production of electron carriers, NADPH, production of replication, transcription, and translation. Replication produces a new DNA molecule containing one strand of newly synthesized DNA and the original template strand. Transcription produces the formation of RNA using DNA as a template. And translation produces amino acids are linked together to form protein. And that's it. That's enough, but go through this a couple times and you'll do okay.